Okay. Thanks, by the way, for that lovely talk. It's really very exciting stuff. I'm going to talk about the evolution that we have seen and that we see coming. This is obviously my view in this realm of activity. And I have got it going. Good. We've heard a number of folks present today on the, what I'll call broadly the power of being able to ask conjoint questions of individual cells in a spatial context to tease apart populations and make this kind of inquiry. And it's sometimes hard to believe, and you have to remind yourself that these kind of tools are really only about, 7, maybe 8 years old. It's really still a technique that's in its infancy. It's become very prominent, and we've seen lots of exciting work come, but it's a very young technology. And so, the reasons why it's exciting, I'm just going to, recap the obvious. There are exquisite tools for looking at various aspects of cellular behavior and disease activity, and being able to combine multiple bits of information on every cell and ask these conjoint questions, is just extremely valuable, and we see pressures that will be inevitable and I think valuable to be able to give this kind of context information across a range of analytes. And so various folks have shown, I think, through their work, and I'm just going to give one more illustration of the value of that kind of information, and this is just a nice way to show it. This is a paper that I think was done by, this image is from a paper done by RIM, as I recall, where they looked at various biomarkers as predicting patient outcome, and you see the first one, the nearest to the diagonal, is just for, kind of IHC. And that's actually the prescribing test for most immuno-oncology therapies today. But you can see that the sort of area between that curve and the coin toss dashed line in a rock curve is really not that much. There's not a great deal of distinguishing going on. And you can see that for some of the RNA and some of the DNA non-spatial results, the area improves. But if you contrast that with what you see in the markers shown in cyan, this is a look at published studies where there's at least one spatial metric, such as density, adjacency, or distance from tumor cells from the invasive margin, a spatial measure of that kind in a multiplex setting. The rock curves are much better. And if you compare the area sort of under the belly there between that and the again, that coin toss line, it's like double. The value of this to guide decision making is clearly enormous. And so, this is one of the main areas that I think a lot of the research and a lot of trends to try and develop tests and guide both therapy development and ultimately patient care delivery, are driven by. So, if that's sort of, I'll say, the motivation, the framing shot, I'm going to talk about, sort of three phases. And I think it's fair to say that the HT, called at the time the Polaris, which is the right-hand box back in 2019, it sort of created the technique or the field as we speak of it now, where you could look at 6 markers on a whole slide basis. You could also look at up to 8 in a counter stain on a field-based region. But it was kind of, really cracked open this to render it from being, I'll say, a heroic experiment to something that could be used in a wide range of experiments. And we saw, proliferation of papers as folks started to explore this and use it in their work. And the other sort of semi-contemporaneous item here is about a year, a little bit more, maybe early 2019, PhenoCycler came along, and I'll talk about each of these in their separate course and how I think the pressures and uses have driven where they're going and where I think they'll take us. It really just raised the ceiling on the plex level. So you could get these very, very rich data sets for exploration purposes. And I remember at this time, folks were skeptical whether you could even stain 10 markers at a time and visualize them. And, the answer is there, scores, 100 are possible. And so, there's kind of a little bit of different dynamics for these two, but I think together they tell us some of the evolutionary pressures, the uses that people are trying to put the technology to suit their purposes, and how I see that driving the field and the tools within it. So I'll start with the HT, and again, I think this was fair to say revolutionary at the time to be able to get more than just a triplex fluorescent reading, and this came really, I'll say, a little bit from the pathologist side, where it was building on sort of tools and workflows that were familiar to that group, and the data sets weren't yet so large as to overwhelm or demand, at least at the outset, new methods of interpretation. But it was a game changer compared to IHC or where you could perhaps look at two things. You couldn't really try and tease apart, your immune cohort, cell state, put in a couple of, landmark markers. So it opened up. But what we quickly saw was that, it was going to be possible to look at these kinds of studies, and you can imagine what the next chapter will be when you are told that you can look at 6 or maybe 8 markers, but there's a pretty hard ceiling there. What I think, at that point, establishing that there was a very firm foundation between the data that came out of this kind of measurement and the ones that people had established and used as, if not a gold standard, certainly the accepted standard for looking at things, which is IHC. And this was really vital, I think, in establishing that one could do quantitative work at a higher plex level with the greater information density, and it wasn't, in that sense, an either-or proposition. So if you sort of picture, that was the world of that era, I'll say 2018 era. The pressures at that time were first, show me that it works, let me get this in a quasi-pathologist sort of workflow. And it was obvious to those of us who'd worked on it at the time, actually at Perkin-Elmer, that this would ultimately have, great clinical and translational potential. And we managed, the three of us who were kind of running that business, to be permitted to spin out that whole division and join with Akoya, where at that time, Akoya had just pulled out of Stanford the PhenoCycler technology, where the ceiling was very, very much higher. And so, to most of you in this room, probably, and many on the attending remotely, this is old hat, but just the notion is that you can put down a cocktail of many, many antibodies, and you can bring in, because of oligomediation between your reporters and your antibodies, you can bring in 3 at a time and read them, and then dehybridize, remove them, bring in 3 more, and so on, and so on, and so on. And so you've read out, a great number, if you wish, of different markers. And so this, again, was very exciting because you could now say, I want to ask actually 15 or 20 questions of all the cells to tease apart and as I look at these sort of conjoint questions. The platform at the time, which I'll whimsically call PhenoCycler 0, because you'll hear me talk about PhenoCycler 1 and 2 in a moment, was really a means to do this on sort of third-party microscopes. And, some of the good things were you could work on FFPE samples. It did give this great, single-cell subcellular resolution indeed. The throughput is not great, run in that way. You can use this stuff on a cover slip format where you could perhaps get maybe 18 millimeters area coverage, and if you image the whole thing, that whole area, it was fairly painful. It would take probably a couple of days, and then the data analysis and extraction would take really a handful of days more. So it was common to feel like, I'll put an experiment and I'll start it on Monday and I'll find out next Monday how things turned out. Really powerful, but again, sort of an experts-only kind of activity. And so we set right away to work at Akoya creating what I think probably most of you who've done work and presented today have used on it. It's this system, the PhenoCycler Fusion. And we really needed to address what were the primary pressures. What was driving the field then was, first let me just answer some questions on one sample at all, and the process of optimization of one's or development of one's panel was fairly lengthy. But it did let one do that kind of work. In Fusion, the notion was, I want to run this on a dozen samples, or 15 or 25 samples, because I actually want to learn now about differences amongst patients and outcomes and so forth, rather than trying to study mechanisms in a very limited cohort of samples. And so the speed became a big focus, and also the ability to work on whole slide sections, and to do that in a way that enabled you to use archival samples on slides if you have them. And so another big element that I think we focused on at that time was to get the data sizes to be manageable. And all these, I think, if you hear a common theme, it's at first it was to sort of say, let me make this technique more broadly accessible to running small studies across perhaps a dozen samples or in that range, where I want that breadth of information, that depth of information. And so it is funny to look at from an engineering side, and it's kind of interesting to think about this as watching engineering and technology build upon a realized interesting technique, and what do you do with it. And if you look at what happened as it went from the PhenoCycler 0, as I whimsically call it, to the Fusion 1.0, almost everything was stood on its head, and sometimes quite literally, like the fluidics were delivered through an open well in the one case, and now they're in a flow cell on a slide. And actually the pump inside the fluidics instrument was actually turned upside down as part of that work, although that's kind of for subtle reasons. But the imaging switched from a cover slip on an inverted microscope to a direct, upright microscope. So again, in order to get some of the performance and ways of working to be more amenable and more open to a broader, less expert, less patient user group, we actually had to turn the microscope on its head as well. And so a bunch of these things went on. Maybe one of the ones that was immediately felt was being able to do the data analysis of each, the sort of image post-processing as it went, so that it was concurrent. And so instead of finding out perhaps a few days later how things went, you could generally look at the imagery within a couple of hours of when the microscopy was done. And so, this is sort of, I'll pause for a second and say, this is sort of, I'll say, getting those two platforms to be in good communication with their first user groups, with their first populations of folks who use them and what they wanted to use them for. And so over the next couple of years, what we've spent kind of culminating in a couple of products that we've launched earlier this year was looking at, I'll say, getting these things to scale so that you could run things more routinely, so that you could run them on larger studies. So when you ask yourself a question like, this would be an interesting thing to put on the Fusion, could I do that for this study within my budget, within my time that I've got? And to try and make these things more practical in that regard. And, kind of the sort of what we've called spatial biology 2.0, I'll pick up with the PhenoCycler. The trends that were driving this were, I like the general format. I don't want to need standing things on their head anymore, but I need more of it. And so in this case, the choice of a number 2.0 is not just a marketing term. It's actually technically correct. There are 2.0 slides in the carrier. And I kind of like that that's true this time. And there were a bunch of other things that were done as well to speed up some of the fluidics operations to do that reporter exchange so that the system can image one while it's doing the reporter exchange and preparing for the next round on the other. And collectively, this set of changes meant that any one experiment completes a little bit faster, and indeed you get two in that faster end-to-end time. And so the kind of implications of this are that now it becomes practical to say, I'm the kind of lab that doesn't run weekends. I actually don't know a lab that strictly doesn't run weekends, but I'm a lab that doesn't run weekends. I can still get 10 slides a week through this thing, and given the large amount of real estate on each slide, I can put a few samples on each one. And so I'm just going to walk through, and I'm doing all this fairly quickly because I think it's probably pretty clear, and I'll take questions at the end if you have them. But the idea was, what we find is sometimes you have a large sample and you want to see it, and you want to look at heterogeneity, and you want to look at multiple structures. There have been some publications that we've seen where, for example, very different tumors present within the same resection. Tumors with very different characteristics present within the same resection. And you want to be able to see that. Folks put large TMAs. This, I think, is a 300-core TMA being imaged in one run here. But another thing which we saw a lot of, and I'm kind of returning to, what do we see? What does that tell me about what people are doing? What does that tell me about what's needed and where I think the pressures are going to drive the field? We were surprised when we started seeing, talking with customers and watching what they were doing with the Fusion 1.0, how many of them were putting 2, 3, 5, 8 samples on a slide. It makes perfect sense. They're trying to deal with the question of a mixture of time economy and reagent economy. I've got this real estate, don't let it go idle. I'd like to get, four samples in my run instead of 1. Very sensible thing to do. And so, we're seeing that pattern, and we've done some things. I'll show one little slide at the end about sort of just some engineering tricks to kind of make that work nicely. But we saw that pattern. And again, it speaks to me of people saying, I'd like to be able to use this technique more broadly, and I'm trying to jam this in. Another thing we see is in the 2 slides, they don't have to be running, the same protocol. You can run 2 separate experiments that have 2 different panels. They can be the same, but they needn't be. And we saw these kind of pressures come in. Folks who were happy about that and said, I'm really glad you had that, because if you didn't, it would have been a headache for me. Because, for example, folks who run core labs and they're trying to get, variety of, I'll say, cats and dogs, through their thing, and they're often judged on their ability to be responsive to their constituents and their ability to get things done, in a timely way. And so their ability to gang different programs within a run. To me, these all spoke of a need for where folks are taking this is to want to be able to apply this more routinely, more economically, and more time economically into a broader range of programs. And some of the things that I'll show just sort of from an engineering perspective that we did were, once you realize that you're going to have a whole lot of different samples on there, you want to make sure that you've detected them all and that you really are going to take pictures of all the right things and focus on all the right stuff before the run begins. And so we found a neat way to image the sample prior to the reagents being applied and up front, to kind of confirm, yep, you got all the ones, or tweak it where we didn't. So, folks could image these more complicated groupings of samples, which often are quite different. Like sometimes you would see, like in a tox lab, you'd see, the usual thing of a mouse where you've got a liver and a kidney and so on and so forth, all from one specimen. But more commonly, we were seeing ones that was just really different stuff from a lab that they wanted to ask of potentially different kinds of tissue, or very, very different from one another. And so being able to make sure that we could identify and do all the machine vision things, we added stuff like this to make sure that you were going to get everything before the train left the station. So again, I'll pause before I pick up, kind of what we've seen as the pressures and the trends on the HT side. And I would sort of summarize it as what I've taken away from this is that people want to be able to have a higher ceiling in their plex level and ask these more complicated questions on a routine rather than an extraordinary basis. And I think it may speak a little bit to what kind of programs have been happening on the HT and which ones folks would like to say, gee, I'd like to supplement that with more information, either in an early phase, in a marker bake-off, or in a routine manner. We see these kind of trends, and my guess is that will drive where we're going. On the HT side, you heard a couple folks talk about how the scanning, that goes fairly quickly. But the way in which we image up to 8+1, or in a whole slide basis 6+1, is through spectral unmixing. We image the sample with cleverly chosen filters and then do spectral deconvolution so we can assign the energy to the proper channel and get rid of any crosstalk, and just as important, isolate autofluorescence and largely remove that from the signal so that you get quantitatively good data. That was done as a downstream process, and I'll have a slide in a sec to show it, but that was brought into the scanning itself. So one can now scan, and while it's scanning, it's actually, so here's in the kind of the workflow of the HT1 was, you would scan, and now in 2.0, we're actually doing the unmixing right within the scanning. So while it's scanning slide number 2, it's unmixing slide number 1. And while it's scanning slide number 20, it's doing slide number 19. And yeah, you do have to wait about 8 minutes at the end of slide, the last, whatever that is. But the overall time hit is very small. And just as important, what we saw was a lot of the pain is that you have to go in and annotate things and sit at a computer and mark them up, and the touch time or the but time is high, in doing that. And so to me, this really spoke of how folks working in these kind of programs, really operational considerations are important. It's really driven by, how much time do I have to spend touching this data to get it to a point where I can analyze it. And this was a big part of it. Another one, which I'll jump back very quickly to show, is over here. We wound up with some new file formats. Back in 2018, it was sort of an open question whether people would accept the idea of storing the amount of data that's associated with an 8-bit, whole slide scan of 8 layers. It's fairly big, might be, 5, 8 gigabytes. In 2023, the question was, I got plenty of space for that. Can you give me more dynamic range? And so what we did was we wound up providing for a 16-bit format so that when the samples were unexpectedly bright, they were not saturated. We could still recover them with full accuracy. And that's at a technical level. What it tells me is at an operation level and what people are doing, it says, I don't want to have to scan a slide twice if this one turned out to be unexpectedly bright. Operational factors like putting them all in and knowing that when you scan them once, they're all done and I didn't have to go back and revise or adjust anything. Those are becoming important because of the kind of work that it's being put to. So if you look at HT, what I would say is I think it really illustrates that, the practicality and simplicity of working in a true translational setting, those kind of factors are every bit as important. I mean, it's foundational that you have good data, but these other ones kind of are really vital as folks try and put them to uses where you may have a large number of slides. This details what happens if you have 500 slides in a study, and I've got several people who are doing ones this size and larger. And literally the touch time of somebody working about 3 months of their life to do stuff that now has been reduced to about 2 weeks. Those kind of changes are really quite material, I think. And so it speaks to me of how in the spaces that the HT is being used, we're really starting to see people getting towards true translational work. There's going to be PI labs, core labs doing research, but increasingly we're finding people saying, I've done that work, I've done some optimization, I think I've actually got a panel, I want to run this across a large cohort study. And so in those cases, it's these other factors become really important, and ultimately I think simplicity of using it, ruggedness and simplicity become the hallmarks. I think, by all accounts on the HT, speed is not the issue, simplification is. I imagine those same pressures will come to bear on the Fusion, 1, 2, 3 years from now. I also think we know that there's, and for probably many of us in this room and many attending the call, we're motivated by the knowledge that the information, if not the direct tests, will ultimately guide patient care and will have clinical, utility. And I think it came up on the panel talk. We're probably a few jumps from that, but we know that that's where we're going. In that world, again, these operational factors become important because you've got X days to turn around a result in a test. And so all these sort of workflow concerns, I think, will become essential there and valued even in things like the translational world. So that's why I think we're going to see, back to this chart, I think simplicity is going to become, from a kind of how do I use it, what kind of sophistication is needed in order to use it, what kind of laboratory can use it. Can a CRO use it? Must it be an academic lab at a top-tier university? These kind of things are going to drive us towards simplicity. There will, I know, be pressures to incorporate other analytes, and I think a lot of the concerns that were spoken of during the panel about need for better data pipelines, maybe different ones for visualization and folks within the lab versus when you get into sort of secondary and tertiary analysis. But I think the one that's going to drive it in terms of making the technique more broadly and more reliably used are going to be ones driving us towards factors of simplicity. So that's my that's my prediction of where I think we're going to see the field evolve over the next 3 or 4 years. And given the tempo that we've seen changes in the last, 7 years, I have a feeling that whatever I predict, it'll happen faster than I imagine. With that, I'll thank you, and I want to thank the speakers today for their time, for their presentations, and everyone for coming. And I'll take any questions if you have them.
Thank you, Peter. Are there any questions? Yep.
Hi, thanks for the talk. I was just wondering, it might be a lot farther down the road than the roadmap you have here, but was Akoya ever thinking about any kind of three-dimensionality to these types of assays? I've seen some cyclic assays go up to something like 40, 50 microns thick, and obviously that adds a whole layer of complexity to things, but getting even that extra dimension could really increase the amount of information you get spatially from samples.
Yeah. So I'll make a little bit of a distinction here. The short answer is probably not as the centerpiece of our work. I'll make a little distinction, which is sometimes you want a little bit more dimensionality just in order to compress it down and make everything still a planar view, just because you wish it were planar, but it isn't, but you're not really deeply seeking to understand three-dimensional structures. My guess is that the latter is probably not going to be a primary focus for us. Making things a little flatter feeling might be something we do kind of in the spirit of simplicity and improving, compressing cells to get better statistics on them, is my guess.
Makes sense.
Thank you. Are there any questions online? Okay, I'm going to ask a question.
All right.
So what you're saying basically is, until now, only specialists could actually operate and engage with the instrument, and now we're making it simple enough that anybody can if they have a question, whether it be research or clinical or translational, they could interact with the system and get viable data that they could use in their publications or how to decide the next step, right?
That's absolutely the story of the last 3 years. Absolutely. And I mean, I'm not going to say we're 100% done, but that's what we're working on. I think we're getting towards where you said.
That is awesome. So spatial biology is a really, really crowded field, okay? And you've been in this field for a while. I'm new to this field. So I ask you, as a person who's seen the evolution of the field from when it was just a few iconoclasts basically doing some assays that would give them more information, to now it's become much more mainstream. What do you think sets Akoya apart?
Well, it would be immodest to say, because I'm quite biased. My sense is that we've got a bunch of people who really understand this, have deep roots in this field, right? And come into that inflection point or that time of great enthusiasm, knowing the ropes, knowing the mistakes to avoid, having the scars. I think this will be my 6th instrument of this kind. So I think that is a big part of it. But to be honest, I think another part is it's always a meeting of a need and a technology that make things go. And I think we were positioned well to do that, and the skills to act on that.
Awesome. Thank you so much, Peter. I really appreciate the time you spent and gave us. Oh, there is one more question. Sorry.
Okay, for my own curiosity, Peter, and because I don't get to have this conversation with you very often, so I've got to like a front row seat to see how we've moved towards this simplicity with the 2.0 versions of our instruments. What do you envision, non-binding, for the 3.0 versions?
Oh man, so I want to be a little cautious because you've said it's non-binding, but everything on the internet lives forever. But I look at this and I think I heard so much this morning about I want content. I want that easier. My guess is once you have content that could be reliably used, I think Dr. Zhang's comments there, for example, are on point. I think once you have content that's available, the next thing is, well, can you prep it for me? So I just put the plate in and go. I think someone also spoke of that in the panel of, I just want the lyophilized kit that goes. I think both of those are going to be active areas to work on. I'd be surprised if the pressures aren't there, and if once you do it, if people afterwards will say to themselves, like, I can't believe we ever did that ourselves, because I think they're natural areas where the pressure towards simplicity, I think, will push us to evolve.
Okay, I have one more question. Yeah, recently I just found out a company, maybe with the name Polaris, is for this kind of automated staining. So is there a common market push for this?
I'll say automating the sample staining is something that I think different folks do different ways, and I think at this point we're glad of any way that people have to do it. We don't have sort of a common party line or endorsement or a favored partnership on how you prepare your samples. There are some nice ways of doing it, and I think that's perhaps one of them.
Okay, great. Thank you.
Okay, any more? Let me go face the people so I don't overlook or get blinded. Awesome. This has been a wonderful, wonderful day. Thank you to all our speakers who made it happen. And it was wonderful content, great questions, part of which was asked by me. But I am very thankful to all of you for being here. And there is going to be networking. There's coffee and snacks outside. And feel free to interact with the speakers. You have any questions that sometimes I don't care how stupid my question is, I asked, but some people, they want to ask questions privately, so please feel free to learn more, and thank you so much for your time and your presence here. Thanks, Peter.